Hello and welcome to the Beyond Biotech podcast number 129. I'm Jim Cornell and this is the weekly podcast from Labiotech. This week we're going over to the dark side, or more accurately, the Dark Genome. Last year, Haya Therapeutics landed a $1 billion deal with Eli Lilly to apply its long non-coding RNA technology to obesity. And we will find out about that deal, what the Dark Genome is, and how it is related to disease and treating disease. Because our guest this week is Haya Therapeutics CEO and co-founder, Samir Ounzain. I wonder if we could start by telling me a bit about the company.
So Haya Therapeutics is a precision, what we would consider next generation biopharma company that's really leveraging RNA for targeting or combating common and chronic diseases that have historically been intractable. And the way we're doing that at Haya is really by focusing on a new area of biology. So we're taking I think you would call it a first in biology approach, leveraging RNAs derived from what some people call the dark genome. So this is the 98% of your genome actually that does not code for protein. You may remember when it was called junk DNA. Our approach is really driving insights and driving drug discovery and drug development to actually start targeting the dark genome to precisely control what we think of as the fundamental unit of common and chronic diseases, which is cell states. So in most of the common and chronic diseases that have a large impact on society, they're essentially caused by your cells speaking to the environment and speaking to the common genetic variation that you have embedded within your genome. And what's become very apparent is the information processing in your genome, how your genome speaks to the environment to change cell states, to drive these disease-driving processes in common and chronic disease. It's not happening at the level of messenger RNAs or protein coding genes. It's actually happening at the level of the dark genome, the so-called 98% of your genome that used to be called junk. And what we've really been pioneering over the last 4 years now is this concept that the RNA from the dark genome can actually provide you with a very precise, safe and effective substrate to reprogram its activity and therefore control cell state transitions that drive these disease processes, one obvious example being fibrosis. So that's ultimately what we're doing at Haya. We're pioneering these RNA-guided therapies to reprogram disease-driving cell states. And the ultimate vision actually of Haya is to enable a world enriched by an extended health span by developing these highly precise, safe, and accessible RNA targeting medicines for these type of diseases. And of course, many of these diseases, as you're aware, are highly associated with age. So many of these common and chronic diseases plague societies, in particular aging societies. And, maybe just the last point I would mention, I think it's fair to say that despite all of the amazing advances and innovations in the biopharma industry over the last 20 to 50 years, common and chronic diseases are still a major unmet medical need. And we think our first in biology approach going after the dark genome can start to unlock real therapeutic applications in these disease indications.
I wondered before we first get into some of the nitty-gritty about what you do, if you could give us a bit of background on long non-coding RNA and the dark genome and what they are.
The dark genome is this what's called non-coding regions of the genome. And what we've understood actually in the area of epigenetics is that the dark genome is the location where most of the epigenetics happen. So we know that epigenetics is central for how your genome speaks to the environment and how your cells change in response to environmental signals. But what actually happened, this was around 2010. So with the advances of massively highly parallel sequencing-based approaches, we were able to start looking not just at all of the DNA in your genome, but we could start assessing all of the RNA produced by your genome. And around 2010 and 11, what became very apparent is most of the RNA produced by your genome is not coding for messenger RNA. It's actually derived from this dark genome, this region of the genome where the epigenetics happen. And these RNAs, the technical term for them are actually long non-coding RNAs. And it's really emerged now, in particular over the last decade, that they are the transactors of this information processing. They bridge the world of proteins with the digital world of your genome in how it responds to the environment. So these long non-coding RNAs, or they can also be called lncRNAs, are really emerging as central integrators and information processors for transacting this activity in your non-coding dark genome. And why is this quite exciting from a drug development or discovery perspective? Well, these RNAs actually are highly specific. So the activity of the dark genome is very exquisitely specific to specific cell states in specific tissues and specific diseases. So what that means is if you can drug that activity, you can have potentially not just a very potent effect because of the biology of these molecules, but potentially a very precision, safe and specific effect due to the specificity of its activity. And these RNAs are conferring that. And what's beautiful about RNAs, obviously, emerged as a really exciting therapeutic biomolecule, especially over the last 5 years, is we now have a toolbox of modalities, genetic medicines, that allow us to start drugging and targeting RNA molecules. So by leveraging these RNAs as a substrate for genetic medicine approaches, we can start intervening and reprogramming the regulatory genome, and therefore reprogramming these disease-driving cell state transitions, having what we believe will be truly disease-modifying effects in these common and chronic disease areas.
What's the connection between lncRNA and disease?
Yeah, so I think there are two clues that really have brought these long non-coding RNAs into focus in terms of causality. So why are they potentially causally responsible for the disease-driving processes underlying these common and chronic diseases? Well, first of all, there's the biology and the mechanisms associated with these RNAs. One of their central mechanisms is to coordinate the activity of, for example, epigenetic factors and transcription factors that control cellular behavior. And we know that cell state transitions are what drive the pathophysiological processes responsible ultimately for tissue dysfunction. So we know that the cells changing in response to the environment drive tissue dysfunction and disease, and these RNAs are central to the biological root cause mechanisms that causally regulate that process. But there are two other features that I think are really interesting about these RNAs, which is more broadly the activity of the epigenome, epigenetics, and secondly, where most of the common genetic variation is found that is associated with common and chronic diseases. So you'll probably remember post the genome sequencing era, there was this huge wave of what we call genome-wide association studies looking for common and also rare, but primarily common genetic variants across the population that predispose you to these common and chronic diseases like heart attack, for example, or other types of common diseases, diabetes, obesity. And there was actually quite a shock in the 2010s that we were finding all of this genetic variation associated with common and chronic disease, but only 2% of these variants were mapping to messenger RNAs and proteins. So actually 98% of the significant genetic variation associated with common and chronic disease, it's not found in the coding portion of the genome, it's found in the dark genome. So essentially we have these two big clues. We know that epigenetics controls how you respond to the environment, and the biggest risk factor for most of these common and chronic diseases is your lifestyle, it's the environment. And we know that the predisposition to how you respond to the environment to develop these diseases is encoded by common genetic variation, which is primarily non-coding in the dark genome. And what we've now realized is the epigenetics and the common genetic variation linked to these diseases, it's actually enriched in these RNAs associated with the dark genome. So I think they are probably two of the biggest drivers of causality and evidence linking this new class of biological targets to these so far intractable common and chronic disease driving cell state processes.
So once you've determined that, I guess, how do you connect that, and how do you connect the dots, and how do you address diseases with that knowledge?
Yeah, so there are two parts to this. The first part, which I think is where Haya has really pioneered with our platform, is the dark genome is 98% of your genome. It's a lot of DNA and a lot of RNA being produced from that DNA. So how do you actually identify those key RNAs, those key long non-coding RNAs that are causal in a specific and sensitive manner for the disease-driving cell state process that you're interested in, that you know, if you can reprogram or intervene, will have truly disease-modifying therapeutic effects. So that is a huge data science, computational biology, and functional genomic exercise, both to curate the dark genome and then to take those essentially high-quality proprietary curations of these RNAs, because just a little teaser, most of these RNAs, I mentioned that they're exquisitely specific to an individual cell type in an individual disease. This actually represents a double-edged sword for drug discovery, because unless you precisely curate and map the activity of the dark genome in given cell types in patients during disease progression, you won't even find these RNAs annotated in any reference public databases. So the first thing you actually need to do is to build your own curation and discover these RNAs in the cell states and indications that you care about. That's a huge effort using multimodal genomic approaches on patient material. But once you've generated the high quality input data that allows you to curate the dark genome, how do you then actually annotate and discover these RNAs? That's a computational exercise. And then how do you triage or identify within literally tens of thousands of these molecules you can discover in any given tissue and cell state? We will typically discover 30 to 50,000 of these that are completely novel in any given specific cell state. How do you then enrich for those that are causal and master regulators of the cell state transition? So that itself is quite a sophisticated exercise using, I would say, cutting edge computational biology approaches and algorithms, statistical methods, statistical learning to understand causality. So that's the first piece of the puzzle where Haya has really built, I think, one of the leading platforms in the world to do that. But once you've identified that target where you have conviction around causality, and it has the features of a good drug target, highly specific to the disease, only expressed in the disease-driving cell state, so features that will render it not just an effective target, but a very safe target, how do you then actually drug it? Well, here at Haya, we're essentially standing on the shoulders of the giants that over the last 20 years have been developing and optimizing genetic medicines, primarily oligonucleotide-based approaches to target RNA. So here we can leverage what's already been developed in a programmable way to design, for example, antisense oligonucleotides, small interfering RNAs, to engage these novel targets, to, for example, degrade them, or to, for example, impact the ability of these targets to interact with proteins to have a disease-modifying effect. So there are the 2 points to the discovery side and the development side that we really have invested a lot of time, effort, and resources, the team at Haya, over the last 4 years to be able to do exactly that. And of course, once we've done that, we're able to quite rapidly start translating these novel targets and these drug targets into preclinical and ultimately clinical models.
And how then did you make the decision as to what diseases or conditions you were going to tackle?
Yeah, great question. So the way we approached this was, what are the unique features of this biology? So the unique features are that these RNAs in the dark genome can precisely control cell state transitions in a tissue and disease-specific manner. And then we asked the question, what is a disease-driving cell state transition that is associated with many, many common and chronic diseases where we have had limited success? And that brought us to this process of fibrosis, tissue scar. So fibrosis is essentially caused by resting, let's say, healthy quiescent fibroblasts in your heart, in your lungs, in your kidney, in your liver, becoming activated to a myofibroblast, which is a disease-driving state, which through the deposition of collagen and other types of biomolecules essentially causes tissue dysfunction. Now, I'm sure you're aware, we haven't had a lot of success developing antifibrotic medicines, despite 1 in 3 deaths being caused by this process of fibrosis. Okay, and the reason we haven't had a lot of success is because the industry has been focused on the signaling pathways and the proteins that activate fibroblasts. Now, many of these proteins and signaling pathways are highly pleiotropic. So think of pathways like TGF-beta signaling. So what happens is you develop modalities to block these pro-fibrotic pathways You give them to patients, and typically these antifibrotic clinical trials fail in phase 2 because of limited efficacy. And the reason efficacy is limited is because the therapeutic window is very small that impacts dosing. Why is the therapeutic window so small? When you're targeting these protein-based targets that are pan-fibrotic activators, you have on-target effects wherever fibroblasts are in the body. So when you connected those dots, we realized that with this biology, we could identify precise fibroblast activators that are only ever expressed, for example, in an activated fibroblast in the heart. So targets that control fibrosis in the heart that are not expressed in any other fibroblast across the body. So what that allows you to do immediately is imagine precision antifibrotic drug development, which we think was a great way to start entering this space of common and chronic diseases. And indeed, our lead program is targeting fibrosis in the heart. We now have novel targets that have been validated in preclinical models that are moving through our development pipeline for fibroblast activation in the lung, so pulmonary fibrosis. And last but not least, the role of fibroblasts in solid tumors and within the tumor microenvironment. So fibroblast plasticity is really important in the tumor stroma and this process of desmoplasia, which is really a burden in many solid tumors, like, for example, pancreatic. So that is how we thought about leveraging the biology, but maybe we'll touch upon it after. Any disease-driving cell state transition can be targeted using our platform. Just last year, actually, we announced our first collaboration with Eli Lilly to deploy our platform in areas outside of fibrosis. So we think there's a huge opportunity for this technology.
You mentioned the platform. That's the EchoHAYA. Could you tell me how that works?
There's a number of components to it in terms of curation, discovering long non-coding RNAs, and also discovering the features or elements within these RNAs that synchronize or harmonize their activity, as you could think of them as logic gates. So what we've actually been able to develop is ways to fine-tune the effect on cell state changes. So by targeting these individual long non-coding RNAs, we can, for example, control the ability of a myofibroblast to revert back to a quiescent fibroblast. However, by targeting the logic gates that synchronize the activity of these RNAs, we can actually have more potent reprogramming effects and potentially not only block the activation of a disease-driving cell state, but increase resilience of cell states. So we could increase the resilience and potentially rejuvenate certain cell states in, for example, the context of an aging-associated indication. So that is essentially what EchoHAYA allows us to do. It allows us to fine-tune the cell state reprogramming effects we want via targeting the logic gates within these RNA networks.
You already mentioned your lead candidate. I wonder if you could go into a little bit more detail on that and how it works.
Yeah, sure. So our lead candidate is a synthetic modified antisense oligonucleotide that has been developed to target a novel long non-coding RNA that we discovered called WISPR. And WISPR is a long non-coding RNA that specifically regulates the activation of fibroblasts in the heart. And furthermore, when you engage WISPR in a dose-dependent manner, you can reprogram fibroblasts. So you can reprogram the disease-driving activated fibroblasts and induce a switching back to a resting quiescent state. And why that is really important is because the activated fibroblasts in the heart, what they do is they secrete lots of collagens into the interstitial space. So they cause an expansion of the extracellular matrix, leading to fibrosis, which leads to stiffening of the heart, as well as secreting a number of signaling molecules that, for example, have detrimental effects on the functional cells within the heart and on the immune cells within the heart to increase inflammation. So you can think of these activated myofibroblasts in the heart as sentinel cells that control the pathological remodeling of the heart that ultimately leads to heart failure. In particular, heart failure with preserved ejection fraction. So this is diastolic dysfunction, the poor relaxation of the heart. Now, heart failure with preserved ejection fraction is responsible for half of heart failure globally. And as you're probably aware, we have limited disease-modifying therapies approved for that type of heart failure. So an absolutely gigantic unmet medical need with very limited options. So what we've developed is an antisense that can block this long non-coding RNA, block and reverse fibrosis in the heart. And we're now translating it, and we'll be hopefully going into a first-in-subject clinical trial in around 12 months from now. We'll be targeting this in the context of heart failure with diastolic dysfunction, initially in patients who are symptomatic for diastolic dysfunction as a consequence of a relatively rare genetic structural heart disease called hypertrophic cardiomyopathy. So this impacts around 300,000 patients in the United States. Prevalence globally is around 1 in 500 people. So it's a structural heart disease associated with genetic lesions that results essentially in diastolic dysfunction and heart failure. And what's been understood is that one of the major disease drivers in these patients is this process of cardiac fibrosis. So we believe that this will be a first in biology, first in class antifibrotic disease modifying therapy for structural remodeling of the heart, which could hopefully rapidly bring optionality for patients suffering with hypertrophic cardiomyopathy, but of course rapidly expand into larger heart failure patient populations, including those patients suffering with diastolic dysfunction or heart failure with preserved ejection fraction.
So is that kind of stopping it in its tracks so that it doesn't get any worse, or is it reversing it to a state that it's like you never had it?
So the first objective will be to block fibrosis progression, but due to the mechanism and the cell state reprogramming effects we've seen in our preclinical animal models, we also believe there is a possibility that we can reverse remodel the heart. So we can induce reversal of established fibrosis within the heart. But the first primary objective is just to block fibrosis progression, because even by blocking fibrosis progression in patients with heart failure, you could have a very significant impact on hard outcomes like hospitalization and death over time in these patients. So that is the first objective, with the potential exciting scenario that we may also see reversal in humans.
Excellent news, yeah. Are there many companies working on the dark genome and long non-coding RNAs?
I mean, I'm really happy to say that over the last 5 years, this field, this first in biology field, there's a lot of peer and pioneering companies that are really trying to develop orthogonal and complementary approaches to targeting the dark genome. As I mentioned, it's 98% of your genome. I would, if I had to put a number of how many of these long non-coding RNAs truly exist, you could be talking about orders of magnitude more than messenger RNAs, so getting towards the million number. So there's huge untapped potential, and I'm really happy that there's now many, many companies entering the space that are using different approaches to target long non-coding RNAs, both using oligonucleotide-based therapies, but also there's emerging approaches using small molecule approaches to interfere with the ability of long non-coding RNAs interacting with the effectors of their mechanisms, which are proteins. I think many of the approaches are focusing in the oncology space and maybe more rare diseases, for example, contexts where haploinsufficiency is a problem. But absolutely, it's becoming an attractive space. There's a lot of, I think, truly disease-modifying options in this therapeutic area. And not only is small companies, but also big companies are starting to pay attention to this space. And as evidenced, I think, by a couple of recent partnerships in this space, both by ourselves with Eli Lilly and also one of our peer companies, nexRNA, who announced a collaboration with Bayer in the oncology space, targeting these long non-coding RNAs.
Can you go into a bit more detail on the deal with Lilly?
Yeah, so at a very high level, as I mentioned up top, our platform really allows us to reprogram cell state transitions. And as you can imagine, when you think about diseases like obesity and related metabolic diseases, these are diseases caused by the environment changing cell states. So essentially what our collaboration with Eli Lilly is going to allow us to do and support them with is really identifying novel dark genome lncRNA targets that can, in a disease-modifying way, influence cell state transitions that could have, you could imagine, next generation or complementary disease-modifying effects in obesity and the related metabolic diseases around that. So we're super excited because we think this is really a real validation of the platform that we've built. We're now able to start deploying the platform outside of the fibrosis arena. And of course, we have one of the really most innovative pioneering pharma companies in the world to work with us and to collaborate, taking this biology into new indication areas like obesity.
It seems like almost the new frontier of medicine.
We obviously think that there's huge opportunity. And I think what's interesting is if you look at the industry in general and unmet medical needs in general, especially associated with a lot of the burden and the economic pressures we're facing with aging populations, common and chronic diseases are becoming very, very important to start tackling today. And I would argue that if you follow the biology of what actually causes common and chronic disease, independent of the direction that you approach it, attention starts to focus on the information part of your genome, where the software is programmed, and that is the Dark Genome. So we think there's really a huge opportunity, especially using the emerging types of genetic medicines, to start making common and chronic disease truly tractable. And maybe what I would just double down on in terms of the advantages of this approach is when you're thinking about common and chronic diseases, you really need three fundamental things in any new medicine. You need the medicine to be highly effective. So root cause biology. What is the root cause driving the manifestation of that disease? So think of cell state transitions. Think of the cell as the fundamental unit of these common and chronic diseases in terms of how it responds to the environment. But you also need the medicine to be very, very safe. You're going to be treating potentially millions and millions of patients. So you need very effective and very, very safe medicines. And last but not least, you need highly accessible medicines. So when you think about our biological target class, we have what we believe truly disease-modifying causality and effectiveness in terms of the mechanisms of these molecules. Their unprecedented specificity, we think, makes them very, very safe targets. And we can leverage modalities like antisense oligonucleotides or small interfering RNAs that scale up and manufacturing and cost of goods are actually turning these genetic medicines into highly accessible modalities for common and chronic disease for everybody. So I think that's what makes this new biological target space very exciting. We can truly start to think about effective, safe, and accessible medicines for these large indications.
And what is the implication for cost in that?
Well, the CMC, the manufacturing and the scale-up production of RNA-based medicines, I think we already saw with the COVID vaccine. mRNA synthesis can scale, and it's exactly the same with oligonucleotides. So the CMC manufacturing processes, I would say, are somewhat more tractable for large-scale patient populations than maybe some of the other types of approaches that are leading edge or cutting edge. So I think what we can bring to the table is first in biology innovation in target space for disease modification, but what we can leverage, so we have relatively high biological risk, but potential transformational biological intervention, but relatively low technology risk in terms of using oligonucleotides, both in terms of safety. Tens of thousands of patients. We have over half a dozen approved now oligonucleotide-based therapies. Tens of thousands of patients have been dosed with oligonucleotide therapies. We know the class-associated safety and PK. But what we also know is that the scale-up on manufacturing in CMC means you could have costs of goods that allow you to treat hundreds of thousands, if not millions of patients in a commercially viable manner. And I think that's what's exciting about this new frontier.
Where are you at in terms of the pathway from where you are now to commercialization, that clinical trial path, I guess?
Yeah, so with our lead program, we're around 12 months from going into first in human. And with the current path, hopefully in 24 to 36 months from now, we're reaching a point where we can demonstrate proof of mechanism for this first in biology approach in patients in the context of cardiac fibrosis and heart failure. That's obviously going to be quite a landmark transformational moment for Haya and we believe for the field in general, truly making this biology tractable in the clinic. But right behind our lead program, we've deployed our platform in other fibrosis areas, and we're actually advancing quite rapidly some of our novel targets in other areas like pulmonary fibrosis and like reprogramming the tumor microenvironment and the stroma in the context of solid tumors. So really our priority at Haya is to be clinical stage innovators, demonstrate to the world in patients that this biology translates, and then rapidly build out our pipeline to exploit this technology and this approach to hopefully bring truly disease-modifying medicines to patients as fast as possible. That's ultimately our goal. Let's get these medicines that are truly disease-modifying and accessible to patients as fast as possible.
Clearly, from what you're saying, the sky's the limit in terms of the number of conditions and diseases that you can tackle with this.
Absolutely. Think of all of those diseases caused by your environment, where all of the common genetic variation is in the dark genome. These are diseases and cell state transitions that are primed for a dark genome focused approach.
Is there anything that you wanted to mention or cover that we haven't?
We've covered most of the bases. It's an exciting time for the field, and we're really excited at Haya to keep driving and try to unlock the potential of the dark genome for medicines in areas where patients are in desperate need. And that ultimately is our focus. Maybe one last thing I would just highlight is the great thing about this approach is we're using programmable genetic medicines. And I think everybody's aware that once you have a target, which is essentially an RNA target, it's essentially digital information, generating a programmable genetic medicine against that can happen very, very fast. So you can really start to think about accelerated timelines in terms of target discovery, development candidate nomination, GLP talks, and transitioning into clinic. And that's an area we haven't touched on in today's discussion, maybe in the future. Haya has deployed and built a lot of data science capabilities, especially using statistical learning approaches, machine learning, to allow us to accelerate these steps. So we're really imagining a future where we can accelerate target discovery and get assets, candidates into patients on a much faster timeline than what we are imagining today. That is the ultimate vision, so we can go faster.
So if you didn't know before, now you know what the Dark Genome is, if it comes up in a quiz. A really interesting subject, and one we definitely haven't heard the last of. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.